Hi, in this video I will talk about fastq files, where to get them and what to do with them and why. When we talk about differential expression, we usually begin with the expression levels for transcript or RNA-seq counts, but those counts are already the process data. The whole business begins with the photo images, which we do not use. The next step after those photo images, this is a static image of microarray and this is dynamic image for Illumina sequencing machine. They are converted into something more usable. In case of RNA-seq, the first file will be fastq file. And fastq is a peculiar file, and it is a multiple file. It aggregates an information about millions of sequenced reads. Each of them consists of 4 lines. The first line of the record is a sort of address of the sequence. It contains technical information, where it is, what's the index and what's the length. The second line occupies 2 lines because the length is so big, and it is the sequence information itself. The third line can be just a plus or something more. In this particular case, it copies the information of the first line. And the fourth line, which is this one, FFF, it is a quality of the sequence call. So for every base in a sequence, there is a quality given as a ASCII code symbol. And then the record repeats. So that's the second record, and that's the third one, and so on, so on. More details you can find in Wikipedia, for instance, regarding fastq format. It will explain you fields 1 again, 1, 2, 3, 4. But it will also tell you that there are many different formats which are generally known as fastq. In other words, fastq is not strictly fixed. It allows some variations in the line's content. If we put all these file formats next to one another, first we have photo images and then fastq, which contains information about sequence, fragment, and the quality. Then, what we are not discussing, only mention, that the first step in mapping, the bump file will be generated. It will not have info on sequence quality anymore, but it will have information about where in genome the sequence is mapped, and information about mismatches to the reference genome, which can be useful. And this gray zone is where we navigate regarding gene expression. If you only live in this area, not visiting any other places, then you will never have access to mapping information, to the SNP information, to the quality information. All this will be not accessible from a gene expression area. That's why you might need fastq and sometimes bump and sometimes something else. So the question is, can we get fastq files from NCBI Geo, since we get gene expression data from there? And the answer is yes, almost, but strictly speaking, no, not in NCBI Geo. But where is it? Let's have a look at this particular case. It's a small data set dealing with some smoking, non-smoking studies and effect on mesenchymal cells. It consists of 4 samples. Even though it is RNA-seq, you can analyze it with Geo2R, where DSEQ2 protocol will be applied. And you can download RNA-seq counts either with a button or downloading this file. But since we want to find fastq files, It's not on this page. It's over here. It's hidden in SRA run selector. If you click on it, you come to a sequence reads archive of NCBI, and at the bottom of it you will find information about the very same samples, control and treated. And if you click on the run of a first sample, for instance, then you come to another page where you might think it is here, but no, it says the file is too big, and sequencing files are real big. Be aware of it. So it's not in this folder, but in this folder. And these two you can download and use it for your analysis and whatever it is. These two files are two versions of the same sequencing file. And you can ask, is it in a fastq format? And the answer is no, not yet. But you can make a fastq out of those files, and I will show you how. And there are 3 steps. First you download, then convert those files into fastq. And fastq is naturally far too big. Then the third step will be zipped to fastq gzipped format, which is usable for all kinds of applications. So click on those two or both and download it on your computer. You still need sra-toolkit, and you can follow this link, which will instruct you what to do in case you work with a Linux machine. But if you are in Mac, and I am in Mac now, I did it differently. I inquired Brew whether they have a sra-toolkit, first like this, and then they say no, you have to call this, and then you will have it. If you do it for a first time, Brew will install it. If you do it for a second time, Brew will ask you whether you want to reinstall it. Then, of course, I would say thanks, no, and we are ready for the next step, which is converting our downloaded files into a regular fastq format. First, you move your files to the proper place on your computer. Then you open terminal in this proper place, and then you write fastq-dump SRA, and then you see that you have it fastq appearing. It takes some time to convert those downloaded files into fastq format, at least 30 minutes on my iMac with the i7 Intel processor. And also pay attention that the fastq file is much bigger than the file you downloaded. And therefore it makes sense to gzip those fastq files. Unlike the compression format used in SRA database, gzipped format is commonly accepted and recognized by functions and commands. Gzip is directly available on Mac computers and Linux computers. To make sure you have it, you can type gzip --help, and then you see that it is responding and saying what it is. And then you can say, what do I have? I have those files, I'm in the right directory. And then I say gzip sra 2753 etc. .fastq, and after a while it will appear in the same directory. I gzipped both files, and the uncompressed file disappears after compression. The sizes of freshly compressed files are quite comparable to those which we downloaded. Now we are ready to answer the question which version of the downloaded file is better for us, lite or normalized. To do so, you have to be able to look inside the file. Note that it is a zipped file, you cannot open it in a text editor. You can look inside gzipped files with the help of three functions: zcut, zless, and zmore. All of them work in Linux, and you can use zless and zmore in macOS as well. To do so, you have to open a proper directory, then open terminal, new terminal, that folder. So I'm here, and then I can say zless sra fastq-gz, and then you see the inside of the file, and by scrolling down the arrow you can continue and see that it's pretty much a standard fastq file where all 4 lines are available: the address line, the sequencing line, which is big, it's a 302, then the plus line, and the quality line. It's all there. To stop it, you can say q and quit, and then you can repeat it for lite.1.fastq, and then you see that the lite version has also address and the sequencing line and the service line, but the quality line is missing. And therefore, if you care about quality checks, then you better do not use lite. That's the difference between lite and normalized versions. Next, we could use fastq-gz file for quality controls. Actually, you must do quality checks. And of course, fastq files are used for mapping, which will end up with the creation of bump files, and tab files, which are used for gene expression analysis. They will contain reads per transcript counts. But it is another story for another time.